Today we will be doing module 5 of your class, and which is on proteins. So in this you will be learning about how proteins, what are the building blocks of protein, what are their functions inside the human body, and so on and so forth. So the good thing about this module, like I said, is well connected with module 4. So you will feel that it's a continuation of module 4. What you will learn in this module is you will be looking at the nature of protein molecules, and you will also be listing some important members, that is just the common proteins. Also to able to distinguish between primary, secondary, tertiary, and quaternary structures of proteins. That's something really important. Discuss several characteristics of enzymes, making sure that we know how different classes of enzymes function, and then list some important enzymes and discuss their function in human body. So a really nice module. I love working with proteins. And just for a little bit background, my PhD was on protein crystallography. So I really feel very well connected working, especially with proteins. So what we did in module 4, especially since it is so well connected, I hope you remember that we talked about DNA and RNA. That is the two different types of nucleic acids. And the DNA and RNA, they carry the DNA carries the genetic information in this, and it is converted to RNA by a process called transcription. What this process takes place inside the nucleus, and then the DNA is transferred outside, and it's translated to protein. So that's where specifically this module begins. So as we go through the information, you can interrupt me and ask me any number of questions. I'll be glad to stop and slow down or repeat a concept that you need me to. Okay. So this is what we learned in module 4 towards the end, how to convert this information. We know that A pairs with our complementary pairing of A is with U and G with C. And so what happens is that if you are, we also know that T pairs with A and C pairs with G. So this is how, using the Chargaff's rule, that's called Chargaff's rule, the DNA is transcribed to RNA. And a sample of this is shown in this right side picture. Let me use a pointer to show that to you. Okay, so here on the right side, you can see this conversion, that complementary base pairing, A pairs with U and C pairs with G, and so on and so forth. Once that is done, the translation which takes place outside is the one that is converting the, the codon, which is a set of 3 nucleotides of RNA to a given amino acid. If you have any question about this particular thing or anything here feels complicated, please do let me know. Also, to look at this is very important from the standpoint that we are just talking about one gene at one time, and you're just being shown one segment of the gene. So you may wonder, like, how big is a gene usually? So a gene could be anywhere from, it could be even small, but let us say a reasonably sized gene could be anywhere from 100 to 200 base pair all the way up to thousands of base pair. That's how big the genes are. So we have so many different genes, 30,000 genes, about 30,000 genes in human body in each cell. And we talked about this before, but let me just see if you remember. So every cell of our body, so we have 60 to 100 trillion cells, every cell of our body has the same exact genomic information, same exact DNA, base by base, everything is identical. And why does the muscle cell and nerve cell or spinal cord cell or brain cell, why do these cells look so different?
Due to the different proteins or something about protein.
Yeah, you're right. Definitely it's because of the different proteins. And it is just that, the whole DNA, every bit of DNA is not really transferred, or the entire DNA does not make protein. Only 2% of it makes protein. and only some of those genes are expressed in one cell, some of the genes are expressed in another cell, and so on and so forth. So that's what makes every cell different. The RNA and finally the protein is what makes it different, not the DNA. And it's really important because allied health is rapidly moving towards, using genomics and personalized genomics. And so it's kind of important to know this little fact. So coming to the main topic, what is protein? Protein is it's a heteropolymer of amino acid. The word heteropolymer, hetero means different, and then polymer means, combination of monomers. So it's a combination of monomers, but it's not like just the same unit. It's not like glycogen or starch. It's a glucose repeating unit. No, there are different kinds of amino acids that come together, and those amino acids will be connected to each other by a specific bond. We're going to take a look. And that's what is shown on the right side. It kind of looks pretty. I mean, the amino acids, of course, are not balls, but they're just represented like that. And each of them have a full name, a 3-digit abbreviation, and a 1-digit abbreviation, which is helpful in different scenarios. So in this picture, what you are shown is a polymer. You're not seeing a monomer, you're seeing a polymer. And in a way, it is an amino acid sequence. So when we talk about amino acids, we have talked a little bit of some of these functional groups in module 1. So it's kind of nice to take a look again. So if you look at, let's start with the 2D structural formula, because that seems the most comfortable and convenient to, to visualize. So there is a carbon in the middle. It is on one hand connected to an acid group, COOH. Oh, well, that reminds us of the carboxylic acid. And then there is the amino group, which is the NH2, amine group. So that is why it's amino acid. So both the groups are present here. Now you can say, what about this CH3? Is it set in stone? Are we always going to have CH3? No, that's where the difference comes. We actually can have different ones. That is why that whole variety in amino acids, why there are 20 different types. And so that is the difference. That's where the difference comes. Now to interpret 1D is really easy. You're just going to combine the information of how many C's, how many H, and how many N and O's are there and put it together. That's the one-dimensional or 1D chemical formula. And then there is a ball and stick model in which you are representing carbons by like a cyan color. There is blue is the nitrogen, red are oxygen, and then there is hydrogens. Okay, so this is the this is what we call as alpha carbon. It's connected to the amino group and the carboxylic group. So the reason the ball and stick model is really important because it helps you visualize that these molecules are all very 3-dimensional. I know on paper we always study in a 2D mode, so it's kind of hard to bring that picture alive, but that's what it is. It is a 3-dimensional space. So all amino acids except for one particular one, that is glycine, they are chiral. Okay. So do you remember the definition of chirality by any chance from module 1 or 2, I believe, sorry.
It's non-superimposable.
Non-superimposable, yes, and attached to 4 different groups. So if you look here, this carbon, of course, is attached to 2 different groups, but what about the remaining? If we replace R with an H, you can see that there will be H on both sides. And if there are H on both sides, it's not 4 different groups. That its presence as an H is glycine. However, if you put anything here apart from H, it will become a chiral molecule just because of 3-dimensionality. You may not be able to visualize it by just thinking of mirror images, but because it's 3-dimensional, you will be able to visualize that. Okay, so let's take a look at this. It's kind of nice to know all the structures. For me, the best way to learn always is to get all the way deep down and understand the inherent structure of everything. And then I feel very comfortable. I'm like, when I'm working with proteins, I can think all the way down. I'm like, okay, made up amino acid, polymers, atoms. I know I know acid, I know charges, I know everything about this. So that's how I like to teach you. There is COO minus, which is the carboxyl group, and it is negatively charged. The NH2 group, it's positively charged because it becomes like sort of, the charge distribution makes it, especially interaction with water, makes it NH3. And then this molecule becomes an interesting molecule which carries a negative and a positive charge, and it is called a zwitterion. And so this is an interesting structure. It has some charge in it, and of course it's an active molecule. And another really interesting thing about the amino acid is amino acids are always L. Do you remember the L and D forms? The D forms are the sugars. So all sugars are D, all amino acids are L. Wow, that's how the nature has, that's how it is present in the nature. And I remember when I read this for the first time, I was like super fascinated. How can one type of entire class of macromolecule be always D and one class of macromolecule is always L? Wow, that's something, because it's not as simple as we think, because these are all right-handed, one is right-handed, one is left-handed forms. And that decides how the enzymes function, like how the enzymes will metabolize them, or how the enzymes will perform some modification reactions. So that's what it is. So yeah, just a joke, amino acid. Okay, so just for you to see. Okay, so it is just the reason it says give me your lunch is because the lunch is for protein, and the protein breaks down into amino acid. Okay, so it has two groups, like we said, the amino group and the carboxyl group, and that's what we just learned about. So this refers to, the term amino acids refers to alpha amino acids, because both amino and carboxyl groups are attached to the alpha carbon, the first carbon, the alpha carbon of the structure. And there are different types though. That's the most interesting thing about amino acids, like the DNA, ATGC done. Everything kind of purine, pyrimidine done. This is not like that. There is a lot of variation. There is a lot of variety. There is, there can be non-polar ones in which are hydrophobic, which hydrophobic, of course, you probably know, it's water, doesn't like water, water phobia. And the polar, which are hydrophilic, which love water. And then there can be different varieties in that. There can be uncharged ones, which are having both kind of charges, but it's overall neutral. It could be charged. It can be anything. It can be any of those things. So let's take a look. What do we mean by all of this? Like, do we have real examples? So yes, we have, and I did list them for you. And I honestly don't remember if I made this up or I took it from somewhere. But you don't need to memorize this. Okay, so the just non-polar side chains, those are grandma always visits London in May for Winston's party. So you can just break up each one of them. Those are all your non-polar. The polar one is Santa's team crafts new quilts yearly. Kind of like that because it's Santa coming from polar. And then electrically charged one are the dragon eat knights riding horses. So it's just literally, easy way to remember. Of course, most of the scenarios, even if you take an advanced course, people won't expect you to remember, but this is a fun way to do it. Now, let's take a look at this whole chart. So the first one is not chiral because it is the same group on what we are seeing as top and bottom. But that, leave that apart, and everything else is chiral. Everything else has a different functional group. And if you see here, this is all the non-polar ones are listed here. All the polar ones are listed here, and the electrically charged ones are listed here. Now, take a careful look, and I'm not going to tell you right away. You should be the one telling me, why are non-polar ones, like, is there something common between all of these non-polar ones? How come these 10 are non-polar? or actually compare them with the polar ones. If you compare them with the polar ones, so any,
Should we be looking at the R group then, like the differences?
yes, only at the green, the rest is same. Good, good thought. Yes, we are only going to look at the green and the purples and these colors and try to see, is there a commonality between all the greens versus,
Oh, there's no oxygen in the greens.
okay.
No, that's not it. Maybe not.
Okay, so you were right. So, I mean, there is only carbon chains in here, right? All carbon chains, okay. What about this? This does have carbon, but then, like you said, it does have some oxygen or sulfur. You see that? Okay, so that means that is bringing the charge into consideration here. Whether you talk about serine or threonine, you talk about cysteine or tyrosine, all of these, and then asparagine or glutamine. So all of these. Now, I want to also bring your attention. So all carbon chain ones are the ones. So you know carbon chain is non-polar. It doesn't really like water. So it's clear that, you know, that's why these are non-polar. But let's take a look at this one. Wow, this has a CONH2. So this has an extra, you know, amine group. Sorry, the amide group. And if you look here, so this is acid. So this one on here, the one with the orange, you know, highlighted it's electrically charged. And the one on the right side, those are the ones having extra. So this is electrically charged by means of having a COO extra, and this by having NH2 extra. So it's kind of nice to get familiar. You can see aspartate, asparagine. The aspartate is the aspartic acid, which has an extra acid, and asparagine is the one which has the extra CONH2, and similarly glutamate, glutamine. Okay, so that, and then lysine and arginine are kind of unique. They have the extra NH2 group, and so is histidine. So also there is something to observe here, that some of them have aromatic structures in them, which gives them like a different property: phenylalanine, tryptophan, tyrosine, and yeah, not really histidine that way, but these are the ones that are aromatic. So these are just the illustrations of different amino acids, and maybe I should have taken you to this page, but exactly shows the same point. All the carbon chain ones are non-polar. All the purple ones here, which have OH and NH2 respectively, those are polar. And then there is some electrically charged. They're either acidic or basic. So yeah, there is, again, I said, like, there is no need to remember, but I can tell you that if you are doing, let us say you're doing a bachelor's in biology or biochemistry, then you would automatically use it so many times. Actually, biochemistry, I would say, that you would remember that. Like, I remember all of these. I never ever have to look what glutamic acid stands for, or if you give me QGH, I never have to look back. I know all of these. I mean, of course, you know, I mean, by training, I'm a biochemist, so I should definitely know this, but it's not that hard, especially given that A is alanine, you know, those are the easy ones, but arginine couldn't take A because alanine already took it. So it's R. And as for aspartic acid is D. So there is trick ways to remember. Similarly, because glycine took G, so glutamine cannot take G, but I kind of like, when you say glutamine, it sounds like glutamine. So it's like Q is a great one for that one. And aspartic acid is D, so glutamic acid is E. So like that, if you can draw in some similarities, it's kind of nice to be able to take a look at scientific language and decipher it. So complete names, 3-letter codes, 1-letter code. These are the ways of representing. Okay, but exam standpoint, like, I really don't expect you to remember these. What I would expect you to remember, if I tell you what is the difference between polar and non-polar, you're like, I don't know. I mean, that's where you should know that non-polar is the one with carbon and polar is the one with charged groups, and you know, such as hydroxy or CONH2. 1 minute, my battery is like draining. Okay, back now. And let us take a look at essential and non-essential amino acids now. So there is, these essential amino acids are the one that you have to obtain from the diet because they are never synthesized. So these are the essential ones. And I mean, there is another fun way to remember, and this is, I mean, not, I didn't do this, someone else made this. My tall vegan friend is watering kale leaves, like that, because, you know, diet and vegan. So that's methionine, threonine, valine, phenylalanine, isoleucine, tryptophan, kale. So these are going by the one letter codings. Okay, so, you know, I mean, these are the different types of diets in which you get different ones. So that's fine. We don't have to worry about that. So these are the rest of the amino acids which you can get from your own diet. And then, you know, I mean, you can synthesize too. So they are synthesized. Do I expect you to remember all of them? Not really, but I would expect you to know at least one or two of them, like, okay, which ones are and which ones are not, at least some examples. And we all know that methionine is the one that starts the, you know, the starts a chain typically. Valine also does, but methionine is the most common one. So it's kind of nice to, that's one way I remember, okay, essential, methionine is, of course, essential is the first one. And then there are a bunch of these, you know, aspartate, glutamate, asparagine, glutamine, all of these are all non-essential. So how are they actually kind of put together? That's always the thing, you know, there is glycosidic bond for carbohydrates. So what bond for this one? What is that combines them together? And DNA had some bonds too. What are those bonds for DNA? Let's try to think. So DNA, there is the bond that combines the two bases, which is the hydrogen bond, and then there is the phosphodiester bonds on the sides. So like that, there's a peptide bond in here, and that is formed by condensation reaction. And this is so common, guys. You should by now start recognizing there is a condensation reaction, water goes out, and it is, I mean, as the water goes out, the CO and H2 is combined to form that peptide bond, okay? And this is the one highlighted in yellow right here. So what I have to say here is we kind of did pretty much the same thing when we're looking at lipids. Water goes away and then they're combining and making that, you know, the triglyceride, yes. And then similarly, you know, in same thing, kind of thing for glycosidic bond. So it's very common kind of reaction, condensation reaction. So how does this actually play? What does it do? Do the CONH bond, does it do something? Well, we are going to come to that. One more thing that I might have missed out here is the end facing N is called N terminus, and the end facing C is called the C terminus. And N is the starting point and C is the ending point. Okay. So now we're going to take a look at how they're joined. So let's try to recognize element by element. I'm going to try to see if we can. Okay. So it didn't quite let me pick. Okay. So you have the C here. Maybe I should have taken another one. Maybe I should have taken a thicker one. So you have the C here. The C is of course attached to an H, and it's attached to a functional group R. But on the other side, you know, on one side is the CO and the other side is the NH. C, and then similarly in the second one, NH is on this side and CO is on this side. And then, you know, it kind of keeps repeating like that. But of course you can see that there is a variation in the structure, but the pattern is same. The colors here, which means the blue on R or the brown on R or green or yellow, they represent different Rs, the different functional groups. And that's what makes these different. If everything was same, it would be like a bunch of glycines together, and that's what it will be. But that's what gives it that special property, because it makes it three-dimensional. You know, all of this is hard to visualize, but it's kind of actually nothing of it is flat. So the question is, did you find this bond? Let's see. Did you find this bond? That's what, CuNH, CuNH, CuNH. It's pretty obvious. It's going to be there between every two carbons. So that was the main point of emphasis here, that to understand that molecular basis and to understand that there are 4 different amino acids, each connected by a CuNH bond. Okay, so how do we name them? So you might just come across the term, or you might have already come across, but you being in allied health, you could come across peptide related terms. Dipeptide stands for 2 amino acid connected together. Tri is for 3. Oligo is for 10 or 11. Poly is for 50 or more. And then, I mean, if a little more, but something which is really, really large, like let's say there is like, I don't know, 200 amino acids. So then it is called a protein. So if someone says to you polypeptide, please don't get confused. It is also a small kind of protein. So it's important to know that peptide word is used a lot till smaller, for the smaller polymers or the shorter polymers of protein, and then the di for 2, tri for 3, which is kind of obvious. Oligo for a couple of them, and poly for many of them, 50, that's what is used. Okay, so before we go ahead, if you have any questions, like I said, feel free to put it in the chat box, or you can unmute yourself, ask, or you can chat to, like if you don't feel like asking everyone, you can text me or Suruchi, and Suruchi can easily interrupt me, and we can get this taken care of, whatever we need to understand. So one amino acid is called one residue. It's a really important point. If you look on the top right corner, and the one amino acid is called one residue, because they may say like this one has like 50 residues. Like what? Shouldn't even talk about residue. What happened? Like suddenly we're talking about residues now. So that's just an interchangeable word. When you start working in a lab, or you do some of those things, and you use some scientific or technical jargon, and that's one of them. So do these amino acids stay like that? Absolutely not. Okay, it is formed like that. It's a sequence of chain of amino acids, and that's your primary structure, but it never stays like that. If it stayed like that, no function in your body would get performed. So what needs to happen? Well, it needs to fold properly. So there is hydrogen bonding of peptide backbone, which can make 2 different kinds of structure. This is called alpha helix or beta pleated sheets. I'm not sure why the beta word is not written here. I'm going to write beta for you. Okay. And so that's that. It's different kinds of structures can be formed. And then they come together and they form tertiary structure, which can be a combination of helix and pleated sheets, beta sheets. And then many of these, 2 or 3 of these, can come together and form a protein. So that forming the protein is not just like whatever, and RNA made it and it's done. No, there is a primary, a secondary level of structure, a tertiary, and a quaternary. And it's not like first secondary happens, then there's a gap, then tertiary happens. No, it's like happening in conjunction, one after another, one after another. And perhaps tertiary starts forming already as the secondary is forming. So these 4 levels of structure, I definitely expect you to remember. This is really, really important. And so for your understanding. Oops, sorry. Okay, so this is an example of sequence of chain of amino acids, and these are joined together by peptide bonds. You cannot see them. These are the peptide amino subunits. This is the abbreviation. This is how it looks like. So for you to understand that, there is an amino end and a carboxyl end. So just let's take a closer look. We are kind of so into the structure. So amino acids chains, they can fold in a repeating pattern. And then you see this bond, so this dotted line, this is the hydrogen bonding. It's really, really important that these bonds are formed, because that's what makes the structures stable. So that's the alpha helix, sorry. Move too quickly. Alpha helix. And whenever you think of helical structure, it's like DNA, like helix. And then there is beta pleated sheet. So beta pleated sheet meant like pleats, like you fold the paper in a certain way for making, origami, like a little fan kind of. That's the beta pleated sheet. And then these are connected by the hydrogen bonds. And do you remember there is hydrogen bond even in DNA? So not uncommon to have that hydrogen bond. So you can see beta strands are, as they are shown as flat arrow pointing towards carboxyl group, and this is how it looks like. Now for tertiary structure, how does it actually form? So like I said, always love to have a little bit of inner perspective, because when I think of protein as a whole, now I'm always thinking of like, okay, they're having all these bonds. So there is hydrogen bond. There are hydrophobic interactions where a hydrophobic R group is like interacting with other hydrophobic R group and saying, hey, I hate water. And he's like, oh, me too. I hate water too. So they have this little hydrophobic interaction. There are disulfide bridges where sulfur can come together. There are ionic bonds. So there are different ways of these chains to interact. And that's how this beta sheets and alpha helix come together. Now you may say, I don't see any beta sheets here. Well, I would say that this is definitely a helix, and anything that is running parallel and connected, those are the beta sheets. That's how it looks like. And hemoglobin is 4 subunits. So this could easily be like a hemoglobin, 4 different subunits coming together. And each amino acid is shown in, each amino acid chain is shown in different color. And what is this? You may wonder, what is this coin-like thing? That's a substrate. Let us say hemoglobin typically, have a certain cofactor or iron or whatever. So that's what it is here. So some proteins, like we said, made up of 1 or more amino acid chain, more than 1, and that's what gives them quaternary structure. So these chains are held together with same force as the tertiary structure, and that's what we have just discussed. So some of these are like, there could be potassium channels, antibodies. You read so much about antibodies, all these different colors, they each symbolize one big peptide. Then there is G protein. This is like 3 non-identical. And this one antibody has, let us see, 4 amino acid chains, 2 short and 2 long. So that's how the structure is. And thankfully, we're kind of almost done with structure, but I'd love to show you this one. We just talked about this hemoglobin molecule, which is present in RBCs. RBCs contain many, many thousands and thousands of hemoglobin, which transport oxygen and to the heme and hemoglobin in the hemoglobin molecule. So before going to the next slide, I'm so excited to share you. I saw recently there is a UMB spin out. The name of the spin out is Kalocyte. That is Kalocyte. It recently got a huge award, and it has many, many funding. And what they make is dried blood. It's called erythromer. What's the goal of this? The goal of this is, can you guess the goal of this? Making dried blood?
No.
Well, when there is shortage of blood, what you're gonna do? So, sometimes there is shortage of blood. I mean, that's the reality, especially for the rare group types. There is like a B negative, whatever. I mean, they may not just get blood easily. So for those groups, it's probably important. And so they're making, so there's this, they have like millions and millions of dollars in grant money, and they run in Baltimore, making that freeze-dried blood, and they're looking at its applications, how to make it better and whatnot. So that kind of finishes that particular part. Now we're going to why are they important in human body. So proteins, they can be used for variety of purposes, for energy, for import export, for structural support. You probably are aware of many of this or have read in like a bio course. They are enzymes. Most of the enzymes, I'm not saying 100%, but 99% or more are proteins. And they act as antibodies against foreign substance, such as foreign molecules, such as antigens. So they can be globular, or they can be like sort of, these are different kinds of shape, or fibrous. So let's take a look at what is globular, what is fibrous. So globular, as they say, is globe, so round, so it's the round shapes. And some of the examples are here. That is the hormones or insulin, there is enzyme catalase, there is antibodies, that is immunoglobulins. So these are the globular. And then there are fibrous. Fibrous fibers, long chain, long narrow shapes, not soluble in water. Those are, you can imagine things that are fibrous. I mean, there is collagen, there is, it's found in different tissues. There is keratin found in hair, nails, and bird feathers. So all of this is fibrous. So globular, fibrous, these are two different types. So what do globular ones do? They, for example, amylase will convert the starch into maltose, because if you don't break it down, then you cannot really use that energy. So it's energy trapped in the molecule. Then there is hormones such as insulin that reduce blood sugar. There is antibodies such as immunoglobulins, especially IgGs, which come in the beginning, and then there are other ones that aid in defense against antigen, and there is hemoglobin. What about functional aspects of the fibrous ones? Well, they provide structures, they help in contractions. You can imagine something which is longer can help give structure, help in contraction, and aids in clotting of blood. So all of these different functions. So this is just a compilation and infographic of what the different roles of protein are. So if we look at this compilation quickly, there is signaling proteins that communicate with other cells. There are proteins in the blood, not just hemoglobin alone. There are proteins that clot blood. There is protein that maintain the amount of liquid in blood, and then there are enzymes, cell membranes. As a part of cell membranes, they're receptors, You might have heard a lot about the ACE2 receptor. ACE2 receptor is the one that actually binds to that COVID virus. So they're important part of some of those mechanisms where how human body interacts with the antigen. And that brings us to the immune system. They are antibodies and fight whatever wrong antigen kind of thing is coming, which body doesn't recognize. And they're activated during infection. They are proteins in muscles. And finally, the structure, different types of structure have proteins because they're pretty sturdy. So yeah, this is, I put this slide for you guys and you can kind of take a look at it. It's like keratin is the major protein component of our hair. So, I mean, does anyone, I think all of you, question should be the other way around. Does anyone never go to the parlor or get their hair straightened? Well, then you're unique, but these days everyone does that. So, you can, a single hair consists of these bundles of these cells and they have these keratins. And so there is a sulfur atom, which kind of forms a disulfide kind of bond. And so there is the distance between sulfur atoms cause your hair to curl. The more the disulfide bonds, the more it kind of curls. So you add a reducing agent and then you can break them and you can kind of, straighten them. Or you can the other way around, you can add oxidizing agent and make them even kind of remake those curves. So the hair perming is all, you know it better than me, I guess, but I mean, it's something which is very scientific based on protein and the disulfide bond. So clearly without proteins, this wouldn't happen. So then we kept talking about in the beginning too, that enzymes are protein, but finally we get to see how. So a protein molecule that catalyzes chemical reaction of substrate, but it itself doesn't get changed in the process. So that is an enzyme. So sometimes what it requires is a cofactor, and sometimes it may require other kind of derivatives, but without enzymes, your body actually will stop functioning, okay? So I think I have an example somewhere later. I think I might have an example. So there are different kinds of enzyme catalysis reactions, and there could be, there are non-enzymatic reactions, which of course don't use the enzyme, right? So what do they use? They use metals. And then there are enzyme-based reactions. So it's not like every single thing in the body or in other microbes and everywhere in the earth is being done by enzymes. No, there are metal catalysts that can also help do certain reactions, but the versatility of enzymes is like unmatched. So enzymes are chemicals with living units that act in the same manner as catalyst in non-living. So catalyst is for non-living and enzyme is for living. Then, so in living units, enzymes are proteins and they're composed of amino acids. They can also be in certain cases based on RNA, but it's not something we're going to discuss. It's very few. Now comes the energy of activation. So a certain amount of energy is always required to start a reaction, and this is called the energy of activation. So what enzymes and catalysts do is they lower the activation energy. So it's almost like, let us say we never hold office hours. So you're like, oh my God, I have to read the module all on my own. I have no one to ask any questions. So that's like so much of energy of activation needed to read. However, if we already do a lecture and then, you can come and ask questions, and that just makes the process easier. So this acts as it's a catalyst, like just lower the activation energy so you can quickly get to your goals, right? So how does this actually happen? So we are going to take a brief look because, again, allied health would use a bunch of enzymes and proteins, and it's important to know how this works. So, and a catalyst will convert a reactant to a product. So does an enzyme. The only thing is the name is different this time. It's called a substrate. Substrate plus enzyme gives a product. But in between that, there is an intermediate stage where an enzyme substrate complex is formed. That's why it is labeled as E and S. So that's the substrate, that's the enzyme. And what happens is that there is a binding step in which they come together. And once that has happened, then after that, the catalysis leads to the product formation. I hope this is clear. And if it's not, feel free to stop me. I can explain it one more time if you need me to. So this is how basic reaction happens. It has an intermediate step where enzyme substrate complex is formed, followed by the product. So nomenclature of the enzyme is based on the substrate that it acts on. So suppose an enzyme acts on lactose, it's called lactase. Maltose, it's called maltase. ASE is the key word, and I think we learned it in the beginning too. If you say ASE, that is a enzyme. Amylase, protease. Okay, so all of those are enzymes. And what do they make? So, or what do they result in? So lactase enzyme makes glucose plus galactose. So this is what we did in module 2. Maltase makes glucose, cellulose makes glucose, lipids. Module 3, lipase converts lipids into glycerol plus fatty acid. And starch is converted by amylase to maltose, and so on and so forth. So this is how the enzyme nomenclature is based on. So next time you see an enzyme, you can say, I kind of know what it might be acting on. If it's a hydroxylase, it acts on hydroxylase. So the property of the enzyme is that it makes the reaction 10 to the power 3 to 10 to the power 17 times faster. And that's like speed, and specificity. It's only one reaction, high speed and mild conditions. So not really hard to achieve. So what happens without an enzyme? Like I said, it takes like forever to do the task of breaking lactose into glucose plus galactose. But with an enzyme, that task becomes small and you can quickly get glucose plus galactose. And that is what happens when you lower the activation energy. It's exactly the role of an enzyme, lower that activation energy. And see explanation on how enzymes lower activation energy. We're going to take a look at that. Okay. So just to give you an appreciation for how important the enzymes in your body is. There are chemical reactions that are essential for creating the building blocks of DNA. And if you see here, it would take 78 million years without an enzyme. So this many seconds, but with an enzyme, it takes only 0.01 seconds. So, I mean, if your body was to wait for 78 million years, of course, like no human being would live that long. So, I mean, essentially, you will not be able to build the block of the DNA. So without enzyme, your body's function is impossible because enzyme which are the protein catalysts, they speed up the reaction by so many magnitude. And so this is important for you to recognize. And I really love learning about the scientific facts where I'm learning about my own self or own human body, and there's so much incredible things happening. Another example, there is carbonic anhydrase. What is the job of this? Carbonic anhydrase. It catalyzes the hydration of carbon dioxide. And, it is one of the fastest known enzymes. So, it processes, like it would process if you have no carbonic anhydrase, it will take one molecule processed in 100 seconds. And now with the enzyme, it can process 10 million molecules in 100 seconds. Wow, what a difference. It's 10 million times faster than non-catalyzed reactions. So these reactions like this and this, like no way your body can function if there is no building blocks, no hydration of CO2. Like those are the things that are important in certain pathways. So I hope that this creates a little bit of appreciation. So I think this is one of the last parts of this unit where we are categorizing them and based on international classification. So there are 6 different types of groups. There is oxidoreductases, which the job is to catalyze oxidation or reduction. So just go by the name, oxidation, reduction of substances. And some of the examples are lipid oxidation or browning, that is, adding oxidizes phenols in food. So those are 2 important ones. Then there are transferases, as the name suggests, it transfer the group from one to another. Like for example, transferring a phosphate from ATP to another molecule. Okay, so that is the transferases. Then there is, so this is not for, transferase is not really for breaking up the food molecules. So the hydrolases, this catalyzes the hydrolysis reaction. So hydrolase, hydrolysis. And this is the most important enzyme with respect to food quality and for use in food processing. So some of the reactions are, it gives, proteases, helps in texture or protein modification. There is amylases, and then there is lipases. So these are all related to food and how the food breaks down. So this cleaves the peptide bond, this cleaves glycosidic, this cleaves ester. So all of these are cleaving between bonds. Then come the next. So oxidoreductases, transferases, hydrolysis. Then there is lyases, which catalyze the removal or addition of chemical groups to certain substrates. And they just, again, not very important from the food breakdown point of view, but it is something that add water, ammonia, or CO2 across double bonds. So these are, or you can even remove them. So it just, that's what it does. Isomerases, you learned about isomers, so it just helps in that arrangement, rearrangement. The fun part, L2D isomerization or different kind of reactions, which kind of just switches around the structure. For example, glucose can be converted to fructose by glucose isomerase. And, that's one reaction example. Then comes the ligases. So I don't know if you've read any of molecular biology stuff, but it's used for combining. Ligase is like glue. It's like combines 2 different substrates. So these are the 6 ones, and these are all summarized here. I really like this table, so I thought I'll put this out here. It shows how reduction oxygen happened, transferase, it clearly shows yellow gets transferred, hydrolases, lyases, how it makes something new, isomerases, just changing out, and then ligases, like joining things together. And here are some of the important classes of those. So what factors can affect enzymatic activity? This is the easier part. Proteins get affected by temperature, pH. What is the substrate concentration? That can affect activity. What is enzyme concentration? That can also change the amount of time it takes. And then coenzymes and cofactors. So how are then enzymes used in clinical medicine? What is the different things? They're used in diagnosis, prognosis. They are used for curing as part of therapeutic agents. They are used in as diagnostic, part of diagnostic reagents. So I mean, kind of, I mean, not really diagnosis and prognosis. First one is like you take out blood and then looking for biomarkers, but they can be also part of reagent. So here are some examples. There is plasma enzymes are of two types. There is functional enzymes synthesized in liver and present in blood in high concentration. So there are some enzymes which are associated with blood coagulation. So diagnosis and prognosis by mean like there is a change in concentration, and that tells that, okay, this change has occurred and this probably can lead to this particular function or this particular condition. Then there are non-functional enzymes. They are present in very low level and they have really no function. They are released in plasma as a result of cellular damage. So if you see a certain enzyme in blood, I mean, the plasma, then you're like, oh, this enzyme should never have been there. Since you see this enzyme, this person probably has this kind of infection or this kind of hepatitis or whatever that is. So how they are used in diagnostics? They are used for diagnostic purposes. Different enzymes are used to detect like different conditions. So there are some of them are listed here. Also, what can happen is that enzymes can have different isomers. There are isoenzymes. Same enzyme can have different forms, and they can be synthesized by different tissues, and they can work at the same exact reaction. Okay, so there are some applications even for those like lactodehydrogenase and creatine kinase. There are different applications for all of this based on what you are trying to find out. The time is now 5, but we are kind of almost, getting done. So just, I think maybe not more than a couple of minutes. So the lactate dehydrogenase, it's a tetrameric enzyme formed by combination of these different subunits, okay, which originate from here. And you can see that these are different types. There is lactodehydrogenase 1, lactodehydrogenase 2, 3, 4, and they have all 4 subunits, but some is H3M1, some is more heart muscle, some is more some is more heart, some is more muscle. Those are the different subunits. And what do we test? Like, for example, LDH is increased in certain conditions. So like that. Similarly, creatine kinase, it could be, you could see levels going up within, and so you can use it within very short time, and you can use it as a diagnostic purpose. They can also be used for curing, and that would be like used in clearing blood clots. They are used in, they will cleave that fiber-like thing so that blood clots can be cleared up, because clotting usually involves some kind of fiber formation. Then asparaginase used in adult leukemia decreases asparagine level, which is needed for tumor cells. Okay, so these are all the different enzymes used for different things. Glucose oxidase is used for glucose estimation. Similarly, uricase for uric acid, urease for urea, and all of those. And what is shown here is a example of how is glucose measured using a glucometer. I'm pretty sure you might have seen this. You might have seen a family member do this. So that's just giving you an example as to what happens and how enzymes help in detection, and then change, that color change is the one that tells us. But actually what is involved is the proteins and enzymes. So what happens in dental hygiene? Well, we know that there is oral cavity has saliva. Saliva is enzymes. Everyone knows that. And so total protein concentration is pretty low. But there are some important proteins that I am sharing here with you. And the main enzymes that are present in saliva are amylase, lingual lipase, carbonic anhydrase, and peroxidase. And some of these enzymes and proteins, including saliva, specific glycoprotein, they're synthesized by particular cells. So this is just all of it put together. Monomer to polymer, nucleotide makes DNA, monosaccharides make starch, glycogen, fatty acid makes a fat molecule, and that is what's a part of adipose and fats. So that's about it for now. Like I said, it will take only a couple more minutes. You're welcome to try out some of these things. And I want to stop recording now.